Part 2 of questions on spermatogenesis and oogenesis. The body of Balbiani, the corpus Balbiani, is present in the... So the answer is primary oocyte. The body of Balbiani is basically distinct and special for the primary oocyte. Here's the primary oocyte, it's round, and here's the spherical nucleus. Next to the spherical nucleus, right adjacent, is this body of Balbiani, which just has the membrane organelles, which just has the mitochondria, the Golgi, and the proteins, and the endoplasmic reticulum is within body of Balbiani. The human spermatogenetic cycle occupies about 72 days. It's a fact. It occupies 72 days. The diameter of human ovum, already you know that the human ovum is quite big in comparison to the sperm. So the human ovum is 120 micrometers, whereas the spermatozoa's length is 60 micrometers, which is half, okay? So here I've tried to illustrate this. Diameter of ovum is 120, spermatozoa is 60. The oogenesis is completed after fertilization. These are multiple choice, and here you have to select the correct answers. Which of the following are specific for the acrosome? Basically, the acrosome is within the sperm, and basically anything within a sperm and acrosome is essentially a membrane organelle. Because this is the acrosome, right? This is acrosome. It's referring to the sperm, so it can't be is built in the primary oocyte, because primary oocyte is all for female reproductive system. This is male. Contains glycogen. Why would the acrosome contain glycogen? What's the role of the... These two are easy, because what's the role of the sperm and what's the acrosome? Basically, the acrosome tries to hydrolyze through the ovum and enter the ovum, so it doesn't contain glycogen. The acrosome contains hydrolytic enzymes. Biochemical analogous to the lysosomes, because lysosomes also hydrolyze. This is true, so this is yes. And it's derived from the Golgi complex. We've already talked about how the acrosome is a Golgi complex derivative. It's a derivative of Golgi, so that's correct, and it's a membrane organelle. Membrane organelle derived from Golgi complex, biochemically analogous. Okay, which of the following is specific for spermatogenesis? It starts during the embryonic period. No, this is for oocytes. Oogenesis starts with the onset of puberty. Yes, it does. Spermatogenesis does start within puberty in men. Passes through 3 stages. No. So basically, oogenesis has 3 stages according to this handbook, and spermatogenesis has 4. The 4 stages are cell multiplication, cell growth, cell differentiation, and cell maturation. Which of the following are specific for the secondary spermatocytes? Secondary spermatocytes we discussed already in the spermatogenesis cycle. Basically, here's when a secondary spermatocyte, primary spermatocyte goes to mitosis. When it goes through meiosis to the secondary spermatocytes, here the genome is halved, okay? So this is going to be haploid number of chromosomes are formed during the second phase of spermatogenesis. That's obvious. Spermatozoa head contains nucleus and acrosome. We know this, okay? Next, basically the consecutive stages of spermatogenesis: cell multiplication, cell growth, cell differentiation, cell maturation. Rewind that if you didn't get it. The ovum is surrounded by zona pellucida and corona radiata, okay? A zona pellucida and a corona radiata as a crown. The main parts of spermatozoa: head, neck, middle piece, tail. We know this. Consecutive stages of oogenesis are cell multiplication, cell growth, cell differentiation. Now, here zona pellucida consists of mucoprotein substance, microvilli of oocyte, and process of follicular cells, contacts between oocytes and follicular cells. Type of male germ cells: sperm-related, spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, spermatozoa. Okay, and here we've got some more questions.